In this video, I'm going to talk about the IMViC. The IMViC is an acronym for the different tests, the 4 tests that are found in this system. I is for indole. The indole test evaluates the microorganism's ability to break down the amino acid tryptophan. When tryptophan is broken down, indole is produced. The M is for methyl red. Methyl red is a test that tests for mixed acid fermentation. If the microorganism does mixed acid fermentation, you're going to incubate it in this specialized media, and then you add methyl red to the tube after, and that's going to cause a red color change if it's positive. The VP, or the V stands for VP or Voges-Proskauer test. This tests for acetone production from fermentation. If it's produced, you're going to add 2 reagents to the tube after incubation, and then a red color change is going to indicate a positive result. And the last one, the C. C is for citrate. Citrate is in the media. If the microorganism can utilize the citrate, it's going to eat all the citrate, and then it's going to start eating or consuming the ammonia in the medium. A base is going to be produced, and that's going to cause a blue color change in the medium. So first we're going to start with the indole test. And there's a couple different ways to do the indole test. You can do the rapid spot test, which is what we're going to do today, or you can incubate it in tryptone broth and add the indole reagent directly to that broth. But we're going to do the quick method so you can see results today. So first thing we do is flame sterilize this loop, and then we're going to add bacteria directly to the filter paper. And I'm going to do a couple different samples so that you can see the difference. So once the bacteria is on the paper, you can flame sterilize, set down your loop, and then you're going to drop or grab this dropper, the DMACA indole dropper. There is a glass ampule in here that you'll have to break, and then you'll have to shake it down in order to use it. When you put the drops on the paper, you should see a color change. And it may be difficult to see at first, so you might have to put it behind a white background. So I'm going to grab a white background here. You can see the top one has turned like an aqua blue color. This one is a positive result. The bottom one is a pink color, so that is a negative result. So positive for indole production from tryptophan degradation, and negative. The next tests just involve tubes. So we have the MRVP broth. We're going to inoculate 2. One of them is going to be for the methyl red test. The other one's going to be for the Voges-Proskauer test. And then we have the citrate slant. And this is what the citrate slant looks like. It's green in color initially. So we're going to transfer bacteria to this media. And we're going to use E. coli to inoculate all of these. So grab some bacteria, aseptically transfer it. And we're just going to continue inoculating. To inoculate the slant, we're going to do this on the side. Again, we're just going to paint up the surface of the slant. So we're going to put those tubes in the incubator, and then we'll come back and check those results once they're done growing. Alright, so now I'm going to go over the results of some of these tests. So we already got the results for the indole test, and now I'm going to talk about the methyl red, the MR test. So for the MR test, we inoculated our MRVP broth. It looks like there's some growth in the tube. And now we're going to add methyl red drops to see if it's a mixed acid fermenter. So we have our dropper bottle of methyl red. And set that down. And I'm just going to add a couple drops. And a red color change is a positive result. So as you can see, this is positive for mixed acid fermentation. Now for the VP test, you actually add two different droppers. So you're going to add alpha naphthol and potassium hydroxide. Now I added these, and then you're supposed to let them sit for 20 minutes, and after 20 minutes you can see that the color change happened. So very similar to the color change that you see in the MR tube, you can see that this is positive for VP. The next test was a citrate test. If the microorganism can utilize citrate, it's going to eat up the citrate in the medium, and then it's going to produce ammonia. And if that ammonia is present, that's a base. So you're going to see a color change in the agar. So you can see our tube here is blue in color, and this was the original tube. So when you compare the original, this is a negative result, and when you look at the actual culture tube, this is a positive result. So you can see the blue color change is positive.